computer. Okay, there we go. Okay, hi, Amy Akers here. I'm excited to share with you today some information about clinical trials and current research projects for cavernous angioma. I'm joined by Dr. Issam Awad, who will be presenting the lecture portion of this webinar. Among his many academic and clinical honors, Dr. Awad is the John Harper Seeley Professor of Surgery and the Director of Neurovascular Surgery at the University of Chicago. I'm fortunate to work closely with Dr. Awad as he is also the founding chairman of Angioma Alliance's Scientific Advisory Board, and he serves as the director of our first designated Clinical Center of Excellence, also at the University of Chicago. Today, Dr. Awad is going to share with us some of his clinical research expertise as we talk about clinical trials for cavernous angioma and two exciting new research projects for which his team will be recruiting study participants. Dr. Awad, welcome.
Thank you. This is really an exciting time because the things we're going to talk about this afternoon are really a new beginning in cavernous angioma research, Amy. So we're
Absolutely.
very excited.
Fantastic. Well, thank you very much. I have got your slides up here and we are ready to hear all about this new CASH trial readiness project.
Right. So this webinar is going to address two separate projects. The first one is called Trial Readiness for cavernous angiomas with symptomatic hemorrhage. So the CASH is CASH. And so trial readiness for CASH lesions, we will explain to you why we do clinical trials, why we need a trial readiness project, what are CASH lesions and why we are investigating them, and how to take part in this particular project of trial readiness. A lot of people are involved in this project and their names are listed, and we will mention individual people's roles during this presentation. So the first issue that I'm going to take really a minute or two to address is why do we really do clinical trials and why are they needed? So the problem with drugs being used in humans is that if the drug doesn't work or if it does bad side effects, that can cause immense problem and harm to the trust in the research integrity, and will be wasting time using bad medications instead of developing truly effective drugs. So it's important for us to know that the drugs that we are using actually do what they are intended to do. And just because a drug works in a laboratory animal like the mouse or the rabbit or whatever, does not necessarily mean that it will work in humans. There are different issues in the human beings and their lesions than in the animals. And the animals only help us to understand a proof of concept that the drug might work in humans. And the best motivated candidates from animal research may in turn out to be harmful in human, or not helpful at all, meaning useless, not causing the effect that they are intended. And the last thing we want as patients is to be given drugs that work in the mouse but do the opposite thing in humans. Now you hear about blinded clinical trials. Blinding means that the doctor giving the medication and the patient receiving the medication doesn't know really if they are on the real drug or on a sugar pill instead of the drug. Why do we do that? We do that because all of us want the drug to have an effect. We take a pill, we end up feeling great, so we want to really know that it worked. We have so many biases, and those biases actually can get in the way of uncovering a real harm of a drug. So therefore, it is important to do these trials with a control so that we know a real difference between receiving the drug versus not receiving the drug. And these trials are done in phases. We call them phase 1, 2, and 3. I'm not going to spend a lot of time on that, but suffice it to say that phase 1 is the earliest phase that tests for safety and feasibility, if you can even take the drug and if it is safe. Phase 2 will check whether there might be the desirable effect. And then drugs that pass phase 2 are taken to a big final trial, which is phase 3, that tests for sure whether they work or not. So the Food and Drug Administration requires at least a phase 3 trial before they approve a label for a drug to be used for a given disease. Next slide, please, Amy. So for cavernous angioma, this concept of CASH, or the cavernous angioma with symptomatic hemorrhage, is really where the money is, no pun intended. So the reason for this is because many of us have cavernous angiomas in the brain. They may have a little bleed in the lesion, but when the lesion bleeds and causes symptoms, so both a new bleed and symptoms, that makes all the difference in the world in what's happened the next year or two. So a lesion that has had a symptomatic hemorrhage is 10 times more likely to have a symptomatic hemorrhage in the next couple of years. So that particular lesion is like the hot lesion. It's the lesion we want to take out, we want to give a medicine for, and it's a big difference between studying these lesions versus the many lesions that are not likely to do anything in the next 2 to 3 years. Therefore, the drug may not have any effect because there isn't much going on in the lesion. So this issue of symptomatic hemorrhage has become very important because it isolates the lesions that we should study first, and they are the most likely to have an effect of a given drug. Next slide. So this next slide shows in a graph form, so basically, of more than 1 million people who have a cavernous angiomas, less than 200,000 have really had a recent symptomatic hemorrhage. And therefore, those are the ones that usually get the surgery, which you see on the left. And we want to try to see if the pill can prevent us from having the surgery. And that would be a great thing. Next slide. So the next slide will show you that we, in fact, have many drugs that are being pursued now for cavernous angiomas at different stages of development. And some of them are in current clinical use for other indications like statins or vitamin D and so forth. And these drugs can be repurposed for cavernous angiomas, meaning used for a new label for cavernous angioma if they work in this disease. Other drugs need to go through a lot of they have to jump through other hoops if they have never been given to a human being before. So there is a different pathway for the drugs that we already have on the shelf versus the ones that we are developing from scratch. And much progress has been made in understanding what happens to a CASH lesion in the next couple of years and some of the outcome issues, so that we can maybe target these CASH lesions with the new drugs and see if we can stabilize the lesion and prevent another bleed, and maybe even spare the patient a second disability or even surgery. Next slide. So this table that you're going to see now is a very busy table. Are we seeing it? Here we go. So this table is a very busy table, but it basically shows us several classes of drugs. So the Rho kinase inhibitors, you have heard a lot about Rho kinase. Rho kinase is a drug that stabilizes the endothelium if you inhibit ROCK activity. And some of the drugs like Fasudil will never be used in humans. There is no pathway to apply them to a human being. So we either will use the statins, which also have ROCK inhibition property, or we will develop a new drug called the ROCK2 inhibitor that will inhibit ROCK activity only in the brain, or mostly in the brain. So it will be a good drug that doesn't have systemic side effects, but will target the lesions in the brain. Now, obviously, the statins we already have in hand, and they will come to a clinical trial before the ROCK2 inhibitor, because the ROCK2 inhibitor will take time to ensure that it is safe in a human being. Inflammatory drugs are very useful potentially in cavernous angioma. There is a drug called Tempol that is being developed and may be used in a human being for cavernous angioma within the next couple of years. B-cell depletion drugs are like the drugs that are being used in multiple sclerosis and in ulcerative colitis to inhibit inflammation, and they can potentially be trialed and tested in cavernous angioma. Vitamin D probably has an effect, but the problem with vitamin D is that everybody takes it. So it's going to be very hard for us to justify doing a trial of starving people with not taking vitamin D while other people take vitamin D. So for vitamin D, we've just given up and we say just take it, because there is no downside to taking it. And if it helps, you may as well have it on board. But these other medicines like B-cell depletion and Tempol, they can do harm. And they will be very expensive. So you're not going to pop those drugs just because they might be a good idea. So they will be tested in clinical trials. Other drugs that you see down below from the VEGF inhibitors and others are really for the future. And some of those drugs can have a very severe side effects, like the VEGF inhibitor. Those drugs, we are not sure that the cavernous angioma community is ready to take a drug that is so toxic for cavernous angioma, but that might change in the future and we might test them. There is the drug propranolol that you have heard about that may also work in altering angiogenesis, and there have been some case studies suggesting that, and propranolol is likely to come to clinical trial at some point if additional hoops are jumped through to show a mechanism of effect. Next slide. So with all these drugs, why don't we just do trials? Let's go, let's do it. So the problem here is because there are some knowledge gaps before we can really stand up and do the trials. And the drug companies and the National Institutes of Health have recognized that there is some knowledge that we do not have right now that interferes with our ability to do a proper clinical trial. So let me explain what some of this knowledge is. If we're going to enroll patients with CASH, we have no idea how many of you are gonna step up to a clinical trial. We don't know that. We don't know how it's gonna play out on the West Coast, at Mayo Clinic, in Chicago, on the East Coast, etc. And therefore, to model a clinical trial and say we're gonna do 100 patients or 200 patients, we have no clue where those patients will come from. And therefore, we have to show that there are CASH cases at the centers that want to do trials in order to be able to say we want to do a trial. Nobody's going to give us $5 million to do a trial if we don't know whether we have the patients to do it. So identifying CASH cases, finding out whether we can enroll them within reasonable time from their symptomatic hemorrhage, and then understanding where is their lesion. The patients that end up in the trial are probably the ones whose lesion didn't get taken out. So are they going to be mostly the brainstem cases, mostly the deep cases? mostly the disabled or the non-disabled cases. We don't know that mix, and that mix is very important, as I will show you later, to decide how many patients we need in a trial. The gender effect is important. The drugs may work differently in males and females, so we need to know that. And there are some other features that can make a difference. Next slide. There are also new ways we can look at the cavernous angioma lesion. So to the far right, you can see the MRI scan with the lesion there in the yellow box. That's what we all look at every day on the MRI scan. But if we want to look at how much the lesion is leaky, meaning is it likely to bleed because it is leaking, there is a permeability scan that we give with gadolinium where we can measure the leak rate in the lesion on a color scale like you see in the middle scan. And to the left, there is a scan that allows us to measure the quantitative susceptibility map, which is QSM, and that is the amount of iron in the lesion measured in parts per million. So the lesion that's more leaky will have more iron in it. That QSM is very exciting because we all want the lesions not to bleed. So if at the end our lesion has less QSM, then the drug may be working. If it has more QSM, the drug is likely not working or may be dangerous. So these biomarkers can be the canary in the mine shaft. They can be the ones that tell us what's going on with the effect of a drug in a proof of concept before we throw all our money on that drug and hold up the whole population for 5 years with one drug. So we could do small trials with these biomarkers and pick the best medication, the most promising medication, and then take that to the phase 3 trial. Next slide. So the next slide is kind of a busy statistics slide, but it really tells you that if we knew how many patients will rebleed in the next year and a half to 2 years, the number of patients needed in a trial is vastly different. So if you have 30% rebleed rate, you may need less than 100 patients in a trial, but if you have 6% rebleed rate, you may need more than 1,000 patients. So it can make a huge difference in the number of patients needed if we know how much they're gonna rebleed and whether the drug will decrease the bleeding rate by half or by a quarter, you know, and we have no idea how strong the drug will be. Will it completely shut down the hemorrhage, or will it only shut 1 out of 2 or 1 out of 4 bleeds? So those types of things will be very important for us before we design a trial and decide how many patients we need in that trial. Next slide. So now I hope you're starting to think like a statistician and you look at these complicated tables. This one tells you about another feature. If we are looking at the biomarker and the change in the biomarker is the same in year 1 and year 2 versus the change is a little bit less or a little bit more, then you need vastly different number of patients. So we have assumed to start with that the biomarker change by a drug will be about 20%. So it will change the biomarker by 20, and that will allow us to have somewhere between 25 and 40 patients receiving drug and 25 or 40 patients receiving a placebo in order to show a difference between the drug and the placebo. That is a reasonable number to be able to test. But in order to do that, we have to be sure that we can all do those biomarkers and we can all measure those biomarkers accurately, whether they are done in Chicago or at Mayo Clinic or at Johns Hopkins or wherever. Next slide. So filling the gaps includes 3 questions. One, what's our ability to screen, enroll, and understand the risk mix of the patients with CASH? Second, are the biomarkers or the bleed rates, are they going to be the same at multiple sites? And can we count them the same way across sites? And then how many of these events are we going to have during follow-up, so we know if we're going to need to follow the patient up for 1 year or 2 years or 3 years to see an effect. The good news is that the whole cavernous angioma research community in the United States has come together. We have no competitors, we only have collaborators. So everybody has come together to try to answer these questions together. And the leading teams that have the most cavernous angioma patients have stepped up to be the first sites in which we answer these questions. Next slide. So without this information, clinical trials could use false assumptions and will likely fail. So nobody's gonna give us money to do clinical trials if we don't have this information. So we went to the NIH and we told them, give us money so we get this information. And they did. So we have about $5 million over 5 years to get this information. And this is how we're gonna do it with your help. Next slide. Next slide, Amy.
Should be there.
So yeah, it's here. So this study will include groups at Mayo Clinic, University of New Mexico, UCSF, Barrow Neurologic Institute, and University of Utah, and also will include the Johns Hopkins institutions and University of Chicago as helping run these sites. So the clinical sites that will enroll patients will be in New Mexico, Chicago, Mayo Clinic, the Barrow in Phoenix, and University of Utah. And the ones that will oversee this data will be at Johns Hopkins, at UCSF, in Chicago, and at the Mayo Clinic. So we have teams that are going to look at this data that is gathered at the multiple sites. Now, why is your doctor's institution not there? It's okay. These are the sites that were able to show that they can participate and do the work. So just because you see somebody in Indiana or at Michigan or whatever, if the team is not a researcher team in cavernous angioma, they cannot really do this work yet. So we started with these sites, but hopefully if we write the rules with these sites, those rules will be open to everybody to join. And still you will have sites that are within a couple of hours travel by plane from anywhere in the US to be able to join one or more of these sites if you want to enroll. Next slide. So the study aims very briefly are, next slide, and I'm gonna go very quickly through this. We will assess how many cases of CASH come to these sites, collect the data on the patients using the same tools, the same website. We'll enter all the data in the website, including how sick the patients are, who are the men, who are the women, how old are they, when did they bleed, and then we will monitor them over the next 2 years to see how many rebleeds we have and also what happens to the biomarkers. So you can only be included if you had had a symptomatic hemorrhage within the prior year. And we will check that very carefully with your doctor and with your data before enrolling you in this. Once you are enrolled, you will get to receive 2 research MRI scans if you are at Mayo Clinic or New Mexico or Chicago, or if you are at Barrow or at Utah, You just got evaluated at the baseline, so we know who you are, and we don't have to follow you for the time being. So depending on what site you are at, we will be asking you either for information up front or for information up front and a follow-up for up to 2 years. Next slide. And then we will be doing some technical work, making sure these biomarkers, iron and the MRI permeability that are produced at Mayo Clinic and New Mexico have the same accuracy and reproducibility and precision, meaning they're perfectly the same tests as what has been done already in Chicago. Next slide. And next slide. These are just some technical slides about how we do this. Let's go also to the next one. So the follow-up will be done on the cases that are at Mayo Clinic, New Mexico, and Chicago, and they'll be followed for 2 years, and we will know if any of these cash cases rebleed versus a lesion doesn't rebleed, and what happens to their functional status during that time. Obviously, if you're going to be watched anyway after a bleed, you may as well be watched with the trial readiness project. If you are going to have your lesion operated on, then that's fine, you know, then you can have that surgery. But many people, as you know, they bleed and then their lesion continues to be followed. And these are the cases that we will want to keep a close eye on, because there will be the ones that we do clinical trials on. Next slide. So this is really a group effort. Our team up at the University of Chicago is a small team that kind of developed many of these techniques, but in partnership with researchers all around the country, we hope to get this project done. So, Emil, this is basically the story of trial readiness.
That's fantastic. Thank you very much. You know, this is really an exciting and innovative project that I think will truly lay the groundwork for future multi-center trials. Tell me, have you started enrolling? And potential participants who are watching this webinar, what should they do? Who should they contact?
Yes, so the project was officially launched in December of last year, but the year one of the project was basically putting together the internet site for collecting data, doing a lot of the MRI preparations, and we're ahead of schedule on that. So we anticipate that actual patients will start getting enrolled in the trial readiness in about August of this year, well ahead of the schedule of year one. So the project will then run for 3 to 4 years after that.
Fantastic, great, thank you. So can you share with our viewers a little bit of background as to why you specifically chose to hemorrhage, or why the team chose to study hemorrhage for the study?
Right, so there are several things we want to change on cavernous angioma. We'd like to have the lesion disappear altogether, but right now we have no drugs that do that even in animals. So it's not a realistic target today. The other issue is, can we prevent lesions from forming? That is a legitimate goal, but that is a lifetime goal. And as we know, lesions form very slowly over time, except maybe in CCM3, where they form rapidly very early. But in most patients with cavernous angiomas, they're not forming new lesions all the time. So the drug is not likely to be testable if you're not forming a lot of lesions all the time. So we're left with the only relevant feature for which we act today, which is hemorrhage. Nobody wants hemorrhage and nobody wants to rebleed. So that's why we're focusing on hemorrhage.
Great, that's helpful, thank you. And just the last question, do we need to complete this trial readiness project prior to beginning any future clinical trial?
Yeah, so phase 1 and phase 2 trials that can be done at a single site don't have to wait for the completion of trial readiness. So we can start single site trials, and I will talk next about one such trial that is starting simultaneously. As drugs become ready, we don't have to wait. So we can do the phase 1 and phase 2 before the trial readiness is completed. But for multi-site trials that need many sites to enter the data, you need the trial readiness goals. You need to walk before you run.
Sure, that's excellent. Great, well, thank you very much. And that sets us up for our next talk. Let's take a short, tiny bit of a break while I pull up the next presentation, and we'll be back in just a second.
Thank you. Is that okay? Yeah.
All right. So here we are back again with Dr. Awad, who's going to be speaking to us today about the atorvastatin treatment in cavernous angioma with symptomatic hemorrhage, an exploratory proof of concept trial.
So this is very exciting because this is really the first time we're gonna give a human being with a cavernous angioma a drug with the purpose of preventing rebleeding. This is something we all have been dreaming about, patients have been asking for, and I will explain to you in the next few minutes why we chose atorvastatin and why this drug will be one of the first to, well, the first to be tested in a proof of concept trial. Next slide. So this trial, it's okay. Let's go to that one with the disclosures. It's important. So this trial will be funded by the National Institutes of Health over the next 5 years. It received a off-label use permission by the Food and Drug Administration because we're going to use statin for an indication other than what it's used for currently. And that is an important thing. We can't take a drug today for an indication other than it's approved without permission of the FDA or an overlying reason that justifies it. And there is no other conflicts of interest related to this research. So nobody's getting paid by the atorvastatin drug maker or anything like that. Next slide. So what I want to tell you about today is why we are using statin as a Rho kinase inhibitor and why we are using it as a drug of choice. The next thing is how QSM and permeability will be used as imaging biomarkers in the trial. And third, describe how actually the trial will play out. And this trial will be conducted at a single site at the University of Chicago, where we already have the QSM and permeability working. It cannot be done at multiple institutions because these biomarkers have not yet been validated at multiple places. They have been very well validated at the University of Chicago during more than 5 years, sufficiently for the NIH to trust us to conduct the trial at this one place. Now, in order to do that, we're going to give travel stipends to patients who are traveling from outside Chicago, and also a small stipend for people who are driving a long way within Chicago to come to the University of Chicago to have the trial-related visits. I will discuss what those visits are, but the rest of your clinical care can continue to be at your own institution in Minnesota or Michigan or Indiana or wherever you are getting your care, or Mayo Clinic or Cleveland, and come to University of Chicago just for trial-related visits, and we will give you the drug and we will collect the data on you as I will describe later. Next slide. So the cavernous angiomas are these bubbles of blood vessels that leak blood. We all know we have them in our brain, and they can occur either as a result of a gene in our body or as a result of gene problems only in the lesion. So either way, it's the same genes that are screwed up. They're either screwed up in our whole body and therefore we get lesions, or they get screwed up in only one spot and we get one lesion. And so this is the solitary versus the familial. They all have the same molecular mechanisms. And this is work that Amy and others have done that showed that before we could make this type of statement. Next slide. Why is that important? That's because some of the drugs we're going to use on familial cases will also work on the sporadic cases and vice versa. So we can include everybody and not discriminate against people because they're familial or they're sporadic. Now here is the important concept of the cache lesion. The lesion that has bled is much more likely to bleed, more than 10 times more likely to bleed than a lesion that hasn't bled. Next slide. In particular, this is true in the brainstem. This slide shows that where the circle is are higher rates of hemorrhage for lesions that are in the brainstem that have bled once. They can bleed as often as 30, 40, 50% in the next year. So those are lesions that are very hot and are very likely to bleed. And as you know, they are the lesions where surgery is the most dangerous, often deferred till a second or third bleed happens before we do surgery. So the brainstem lesion that hemorrhage is a very good lesion to give a medication for in the hope to prevent another bleed. Next slide. So this slide shows the complication rates in brainstem surgery. So after brainstem surgery, 50% of the patients are worse off after the operation than they were before the operation, at least in the short term. Then they do it because we don't want the future bleeds, but they are taking, all of you are taking a huge risk up front, having surgery on the brainstem. And we would love to prevent a third or 20% of these from coming to surgery. Next slide. So the biology premise here is that in the gene aberrations or gene screw-ups that are caused by cavernous angioma, you can go to the next couple of slides, it'll all lay out. So when that happens, there are a defect in the endothelial cell, and that defect is mediated. It is caused, the switch of that defect is called Rho kinase activation. So if we block Rho kinase from activating, the blood vessel will be less leaky and the cavernous angioma will be less likely to grow or bleed again. And we have shown that in the mouse models where we give ROCK inhibitor drugs and they will basically shut down the lesion. It prevents the lesion from growing or bleeding. Now, many of these ROCK inhibitors are not going to be ready to be given to patients for several years because they're being developed from scratch, except for the fact that atorvastatin, which is a very powerful statin called Lipitor also, atorvastatin under one drug name is Lipitor, but the generic is atorvastatin. That drug, at the high dose in humans, it has a significant ROCK inhibition effect. Normally, we don't take it for ROCK inhibition. We take it to decrease cholesterol, but it also inhibits ROCK, and that's why it causes the atherosclerosis plaque to become stable. So if atorvastatin does to the cavernous angioma what it does to the atherosclerosis plaque, then it will be a very helpful drug. And in the mice, next slide, we have in fact shown, this is a mouse brain on the left that is on placebo, and the mouse brain on the right received atorvastatin. I'd like to have my lesion like the one on the right rather than the one on the left. And on the lower slides, we see those blue dots in the placebo case that indicate iron or bleeding, which are inhibited dramatically in the mice that received atorvastatin. So this type of data in the mice was very, very promising when we give the mice the equivalent dose of Lipitor 80 or atorvastatin 80 milligrams per day in humans. So that dose is already approved by the FDA. Gazillion patients around the world receive it. I don't know if gazillion is the right number, but thousands of patients receive it and do well with it. We do not know if cavernous angioma patients will do well with it. And that is why the trial will need to be done. Next slide. So the trial will test whether atorvastatin in the cache lesion, next slide, let's see the contents, will change the iron content of the lesion, probably, meaning if atorvastatin is helping humans like it's helping the mice, the lesion in the human will have less blood in it than the lesion in the patient receiving placebo. If you take atorvastatin and we have more QSM, then it's bad. We don't want it because then it's causing more bleeding in the lesions. And this biomarker is like the canary in the mine shaft. We don't have to wait till you have a stroke to know whether the drug is harmful or helpful. We can tell if it's having an effect on the lesion, and that is what we call proof of concept. Next slide. So this is QSM in more than 100 patients who didn't bleed, and their QSM stays the same. In fact, decreases a little bit over time, but it's generally extremely stable. However, to the right, in a lesion that rebleeds, the hemorrhage increases in the lesion and it's measurable. So that difference between baseline and follow-up is what we want to change. Next slide. So this is a curve that shows that this is very significant and specific effect. Those that change bleed, those that don't change don't bleed. Next slide. Now the other biomarker is this permeability biomarker. So we give gadolinium and we measure how much leak there is in the lesion and in the brain itself. And we have already found and published that people who are taking statin for their heart or for cholesterol, in fact, have lower permeability in their brain. And we showed that in cavernous angioma patients. So we know that this drug, atorvastatin, will decrease permeability in the brain of cavernous angioma patients. And we want to know if that change is truly significant between the people getting atorvastatin and the ones that are getting placebo, because it could be like the blood test that tells you you've gotten enough drug. So if we give enough drug that it causes the effect in the brain, then it's doing what we want it to do. Next slide. So in this trial, we will measure both QSM and permeability. So who is enrolled in the trial? So the patients will come to the University of Chicago. Many of them will be from the Chicago area, but many will be referred by Angioma Alliance or from throughout the country. They may keep their own doctor and just come to the University of Chicago for the research studies. And the patients will come and will be randomized. Half of them will receive a sugar pill. The other half will receive a pill that looks exactly the same, but that has the atorvastatin in it. You won't know and I won't know what you're getting, but we will figure out what happens to you over the next year or two, and then we will break the blind and find out if the people on atorvastatin were better off than the ones that weren't on atorvastatin. That's how the trial will play out. Next slide. So you have to have a symptomatic hemorrhage within a year. So don't call me and say, I want to do it so I don't get a hemorrhage. We're not going to do that now. So for now, if you have had a symptomatic hemorrhage and your lesion did not get taken out, like it's in the brainstem or you're thinking whether you want to have surgery or not, you can get enrolled in the trial. And if you ever rebleed, you can still have surgery. So if you're waiting for a rebleed, you may as well get the benefit of the drug while you wait. So this, we think, is going to be very helpful for many patients and doctors that are on the fence. Should I do surgery now or wait? This gives you a little something to do while you wait that could help you so you don't ever have to get surgery. Next slide. So exclusion criteria: you can't be pregnant, you can't have atorvastatin already in your system or any other statin, so you couldn't have taken statin in the past year. So if you popped statin because you thought it was a good idea, you can't be in the trial anymore. It'll confuse us. Or if you were on statin for cholesterol indication, we're not going to take you off and have you get a heart attack. So you're going to be on it, and you're not going to be in the trial. You're getting the drug, and we'll see if it's helping you or not. But basically, we will make sure we enroll the patients in whom we can test the effect of the drug. Next slide. So this is a very good slide that let's lay it all out. What's going to happen to you? So initially, you will get a phone call from our team, or you can work with our team by email for initial screening, during which we will understand whether you're eligible for this trial before you schlep all the way to Chicago. Unless you already are here and you want to come and see me and talk about it, you can do that too. But the first visit is done in people whom we think might be eligible for the trial, and then you sign the consent because you're eligible, and you come here for the last screen to make sure that you are in fact able to enroll, and you sign the consent, and you get a whole bunch of tests at this first visit in Chicago that are your first research MRI, blood tests, etc. And you get given a pack of medications to start to take once a day, along with a log. You have to log what you are taking, because we have to be sure you're not cheating and not taking the medicine one day, taking it another day. So we will ask you to keep a log of your medications, and we will count the pills and do all of that stuff to be sure. And we're also testing your blood to know if the drug is in the blood. So if you cheat, we'll know it eventually. There will be a few phone calls and some labs that are done locally by your family doctor to make sure the statin is working well without causing any side effects. You will get regular calls every few months from our trial nurse to see how you are doing. Those things don't cost you anything, and you don't even have to come to our place. You just have to take our call. And then at 1 year and 2 years, you have to come to Chicago to have this battery of tests done again at year 1 and 2. Now, what happens if you rebleed during the year? If you rebleed during the year, you can either come to Chicago and we'll figure it out together like we usually do, or have your doctor do it locally and let us know. And we will stop the drug after we confirm a rebleed, but you will stay in the study, and we want to know what you are like after a year or two, even though you may have rebled. So we will stop the medication but continue to follow you. And also, if you end up having surgery for your lesion, obviously you'll stop the drug, and we will continue to follow you. If you end up deciding to discontinue the study and to stop the study, you can do that at any time. But we obviously beg you up front that if that's what you want to do, please don't even start the study, because it's bad for the research to start and then not follow through. So if you're gonna, if you know up front that you're not gonna follow through, we'll probably ask you not to even try it. You know, this is not about trying whether you like it or not. It's about a commitment to work with us to see if this works or not without harming you in any way. Next slide. Yeah, next slide. So the statistics I'm not going to spend a lot of time on. but basically we're gonna measure what happens at the beginning versus year 1 and at year 1 versus year 2. We're gonna plot these graphs, and the goal is to make you like the black cases, not like the red cases. So we want the drug to make you not increase your QSM. So the goal here is to make your lesion not leak. Next slide. So we did sample size calculations, and we think we need 40 patients in each group. So patients total over 3 years of enrollment. So it will be up to 5 years of follow-up, since each patient is followed for 2 years. The last patient will get enrolled in year 3 and followed for 2 years. The 40 cases allow for a fraction, about a third of the patients, to have missing data. They couldn't travel, they bled, or something happened, and yet still find an effect of the drug. And we will be able to detect an effect as little as 20%. So if the effect of the drug is better than that, like 25% or 30%, we may not need to enroll all 80 patients. The study will stop prematurely because the effect is so strong that we don't need to keep going. And wouldn't that be great? Whether good or bad, if you know the answer, the sooner you know the answer that this drug helps or harms, we will stop the study and disseminate this information so we can go to the next step after that. So a 20% effect size requires 80 patients for the trial. Next slide. Next slide. I think we're good. Let's see what we have at the end. Yeah, so this is important. So halfway through the trial, we will check and see how we are doing. And if it looks like the drug is not having any effect, meaning it's futile to keep going, we will stop the trial. If it looks like the effect is so strong that we need less patients, then we will adjust the sample size so we finish the trial with fewer patients. So we made those statistical plans ahead of time, and we will live by them. There are also interim analyses for safety that we will do. And if there is any hint that the drug is causing harm, there are hard stops and red lights that will go up that will stop the trial. Next slide. So this includes, again, even though it's done at the University of Chicago, it includes a huge amount of collaboration from colleagues all around the country who are contributing different aspects of this trial, the statistics. Dr. Fleming at the Mayo Clinic is going to oversee the safety, and she is not involved in the trial. So this is important to have somebody outside Chicago who is looking at the safety. So we have done this so it meets the highest standards of rigor, and that the answer will be believable and will carry us to the next level. So I think this was the slide. Let me see. Oh no, the next slide is just about how to enroll. And basically, you just call us at the University of Chicago. This is very simple. Those of you who contact us, you know, you call us anytime, you email us anytime, you'll hear from us within a day or two, and we will get you swinging to find out whether you're eligible and how the trial might work for you, whether you live in Chicago or away from Chicago. Thank you very much.
That's fantastic. Thank you very much. Appreciate your time and your energy and your efforts. This is exciting news, and we cannot wait to fill this trial for you. Let me just ask you I have 3 more questions, and then we will we'll close up here. So first, can you tell us a little bit more about trial phases? You've termed this as phase 1/phase 2A. Why is that?
Yes. So the reason it is phase 1, 2A is phase 1 because it's the first time we're giving atorvastatin for cavernous malformation patients. So we need to know that the minute we start giving it's not going to cause bleeding in these patients. So there is a phase 1 effect that could stop the trial very quickly if we see any sign of harm. So that's the phase 1. The phase 2A is the proof of concept. Is there a signal that the drug is doing what we want it to do, which is the decrease in QSM? And if that signal is true, then we will model a trial that actually prevents rebleeding, which is really what we want. And that will be the phase 3 trial that will follow this. So the go, no go here will be either forget about atorvastatin or let's take atorvastatin to the next level.
Perfect, thank you. So statins are very, very common drugs. Lots of people take them. I suspect there's folks out there who might just want to give it a try. What do you advise to your patients?
Yeah, so patients who have seen me have heard this song already. So giving something a try, you can do whatever you want on your own, but as doctors, to be able to tell you whether it helped you or not, we cannot do it with an N of 1. So if you took the drug and you bled, we have no way of knowing if the drug made you bleed or if you are going to bleed anyway. Do we blame the drug? Do we not blame the drug? So it doesn't tell us anything about the helpfulness or not. So if you're the type of person who doesn't care about finding the answer, you just care about taking it or not taking it, maybe talk your doctor into it and maybe they'll do it, maybe they won't. It's not likely that a good doctor will prescribe statin for cavernous angioma because the data is not clear that it doesn't harm. So I am afraid of giving it without a clinical trial because I want to know the answer before I can start prescribing it. And I don't want to be giving it to 20 people and find out later that I was doing bad things. So the quickest way to find the answer is to do the trial.
So talk about those risks of harm. You know, people often ask us about chances of statin treatment maybe causing strokes or that lowering cholesterol too low might worsen symptoms. Why do you think that this is a good drug, a safe drug for our patient community worth trying?
Yes, so we know that statin will lower cholesterol. So if your cholesterol is already low, lowering it further with statin does not do any harm. We have now several studies that showed that. So I'm not worried about your cholesterol being too low and that is a problem. The main problem is gonna be whether for some reason statin makes the cavernous angioma in human beings bleed more than without statin. And there may be a fear of that. In some studies, there was a fear that statin could cause bleeding in the brain, but yet many other studies found that it didn't. We did a lot of careful studies in mice. The NIH made us do a lot of studies before allowing us to do this trial. So we think we're going into it with as good evidence that it's going to be okay as possible, but you don't know for sure till you try it. The other interesting thing is we have a database of people who have been on statin with cavernous angiomas. So they've taken it for their heart, you know, and for cholesterol. And when we looked at how often those people bleed, they don't bleed as often as the ones who are not on statin. So it was giving us, again, a good cause to think that maybe statin is doing the right thing and is not doing any harm. But again, those cases were not matched, and there can be a lot of bias in this, so only the trial will allow us to get to the answer quickly.
Perfect. Great. Well, thank you very much. You've answered all my questions and shared tons of knowledge with our community, and I really appreciate your time and effort and energies towards this fantastic work, and we can't wait to fill your trial.
Thank you, and anybody who wants to learn more, our team here at the University of Chicago is ready to share with you the information. If you're part of the trial readiness at the other sites, your doctors there will explain that to you as well. Everybody is energized.
Wonderful. Well, thank you very much.
Thank you.